We have the histology of the digestive system, and we are starting here with a view of the esophagus sample. However, we're going to start with just the major tunic layers. So starting on the internal space of the esophagus, we have one, two, and three subdivisions here that together make up the tunica mucosa. And now once we cross, you see there's one, two, and three subdivisions that make up the tunica submucosa. Once we cross all three, that puts us into the area of the tunica submucosa. Deep to the submucosa, we can see that there's these two divisions. There's one, and then there's a second one here. So these two sublayers, and we'll number them, together make up the tunica muscularis externa. And then the fourth tunic is anything that we see here on the edge, not really clear from this illustration. And since this is the esophagus sample, this fourth tunic is called the tunica adventitia. So we have the four general tunics: mucosa, submucosa, muscularis externa. And in the case of the esophagus sample, the fourth layer is called the adventitia. So now let's go into the details specifically for the esophagus slide. Looking at the tunica mucosa, the first sublayer, and this is the trait to look for, where we have the stratified squamous epithelium. That's the trait that we're only going to see for the esophagus sample, and that's the trait to recognize that it's the esophagus. We have this sublayer here. It's not listed as part of the histology, but it's good to point out that there is this sublayer between here called a lamina propria. And this separates the epithelial layer, the lamina propria, second layer. And then the third layer here is the muscularis mucosae. And it's all three of these together, the epithelial layer, skipping over, and then the muscularis mucosae, all three of those together make the larger tunica mucosa. So once we cross the muscularis mucosae, we now must be in the tunica submucosa. And you can see the transition in tissue type and appearance. The third general layer is the tunica muscularis. The two subdivisions we're not concerned with. And then the fourth tunic in the case of the esophagus, and we know it's the esophagus by the stratified squamous. The fourth layer is the adventitia. So just another look at the esophagus sample, just a different esophagus sample. We're seeing here the stratified squamous epithelium. Again, we have that second subdivision. And then here is the muscularis mucosae, making the entire superficial surface a tunica mucosa. And then we're into the area of the tunica submucosa. We now have the tunica muscularis externa. And then at the very edge, the tunica adventitia. So now we're viewing a stomach sample. In the first trait, the ruga. So here we see two large folds. Here's just one ruga that's highlighted. And that's the trait to let us know we're viewing the stomach, the large mounds of the rugae. So again, with the tunic layers, we can start at the very surface. And at the absolute surface, we're now going to see simple columnar epithelial tissue. Simple, just a single layer. Again, there is that second subdivision for the tunica mucosa. And then we see the pink line here, the muscularis mucosae. So making this entire bracket the tunica mucosa. We see a very wide open submucosa all the way up into the ruga as well. We have the tunica muscularis externa with its two subdivisions. And then the absolute edge, in the case of these remaining samples, the fourth tunic will be called the tunica serosa. So we'll zoom in on the stomach. Here you see another big fold of verruga to identify the distinguishing trait for the stomach. The tunica mucosa, tunica submucosa, and now we just see a partial section of the tunica muscularis. So here we're zooming in close enough to start to see the simple columnar epithelial tissue, just a single layer. We have to cross this very wide lamina propria to point out, of course, the location of the muscularis mucosae. And all of this is the tunica mucosa. So now here for the duodenum, we see now distinguishing trait for the duodenum, that first term, the villi. We're looking at these slender spikes here. And lining right on the surface of the villi, we're once again going to see simple columnar epithelium. We have to cross the area of the lamina propria to reach, again, the muscularis mucosae. So making all of this the tunica mucosa. We then cross the muscularis mucosae to reach the area of the tunica submucosa. And we see the transition now to the tunica muscularis. Leading with the very edge as the tunica serosa. Well, here's just another view of the duodenum again. So the distinguishing trait, the slender spikes of the villi. Here it's difficult to see the muscularis mucosae with this wide zoom. The tunica submucosa, all of this area, and you can see the separation, the two distinct subdivisions here for the tunica muscularis externa, and the very edge of that, the serosa. So zooming in much closer now, again, we have a villus, this big spike, and we can see lining right on the surface of the villus is where we have the simple columnar epithelial tissue. And an even closer look. So a single layer, simple columnar epithelium. And in this case, we could even see the trait of microvilli. So the last sample for the digestive tract, we're looking at the colon sample. And a distinguishing trait for the colon is taking a look at the tunica muscularis externa. So that's in here. And we can see that first layer of the tunica muscularis externa. The muscle layer has this distinct appearance, looks like kind of these vertical strips. And that's unique to the appearance of the colon histology, that tunica muscularis, specifically the first sublayer has that appearance. So starting from the luminal surface from the inside, there's the tunica mucosa that once again will have a layer of simple columnar epithelium. Can't quite make out yet. we have to cross the area of that second sublayer. And then you can start to see that line right across here. Again, we're at low power, but that's the location of the muscularis mucosae. So once we cross that, we've now passed through the tunica mucosa, and we're in the area of the tunica submucosa. Once again, you can see there's the transition in tissue type from primarily connective to primarily muscle tissue. And we're in the tunica muscularis with that distinct appearance of the circular muscle layer. And again, we're not seeing clearly the very edge with the location of the serosa, the fourth tunic. And a closer look here on the colon. So we can see the simple columnar epithelium. And now that we're zoomed in close enough, we can see another distinguishing trait of the colon is just the abundance of these goblet cells. These are mucus-producing cells, and you notice they're right alongside the simple columnar epithelial tissue, and they're just in great abundance in the tunica mucosa of the colon. So next we have the sample of the liver, an accessory organ of the digestive system. And here in the liver, we're seeing the space here in the middle, appropriately named the central vein, in the center of each lobule. Hepatocytes, hepato meaning liver, hepatocytes are liver cells, and the rows or columns of liver cells here stained in pink, that's the term hepatic plate, are the rows of liver cells, hepatic plates. Whereas a sinusoid or a sinusoidal capillary, these are now the spaces appeared here in white that run between hepatic plates. So all around the sample, we see a central vein in the center, and there's just multiple sinusoids, just the small white canals that are, of course, connected to that central vein. Capillaries, sinusoidal capillaries connected to that central vein, lined by the hepatocyte cells. And a closer look, central vein, and now we can clearly start to make out the nuclei of the hepatocytes. So the row of hepatocytes called hepatic plate, and the spacing between them are the sinusoids. And the last sample, another accessory organ, the pancreas. And the pancreas has a collection of generally two types of areas. We can see all of this area here, and you notice the cells have a lighter stain to their cytoplasm. This entire area is known as a pancreatic islet, as opposed to, you notice the darker majority of the tissue. So the cells that make up the majority of the pancreatic tissue with a darker stain to the cytoplasm. But we have the pancreatic islet, and in contrast to that, what we want to see and focus on now is the pancreatic acini. So it's this portion here, the darker stained majority, that's involved in the digestive system. So that pancreatic acinar cell, making those groups, the pancreatic acini, the digestive portion of the pancreas.